Hi, all of you. Today we'll be moving on to the next method, which is the cytogenetic method. Yesterday we discussed about the pedigree method, how it is useful in discovering the medical histories of a family, of a person, and how it is useful in detecting the genetic diseases or genetic abnormalities that are prevalent within one generation or within one whole family across different generations. So using those records, we were able to trace back different genetic diseases prevalent within one family, right? So today we'll be moving on to the next method, which is called the cytogenetic method. It's generally used along or generally referred along with the biochemical or the molecular method, but I'll try to segregate all these three for your further clarity, because when you talk about molecular method, for example when we talk about cytogenetic methods, these specifically talks about chromosomal abnormalities and how we try to study the chromosomes. When we talk about molecular methods, we try to and talk about polymorphisms at the molecular level, right? And then the biochemical methods. So all of these have to be very segregatedly studied. So I'll try to bring out lot many examples along with these methods as we'll go simultaneously, right? So firstly, why is it important to study cytogenetic method? Now, so far we have studied a lot many things about medical genetics, or how is it being applied in the study of human, its applications in terms of human health, or studying genetic disorders, or even new genetic innovations, technical wise. So understanding this role of genetics in medicine and its applications altogether is altogether the idea of medical genetics, but using that to improve its applications, that baseline understanding or the significance of how we are understanding cells at the at the basic level, how we are trying to study the genetics at the cell level, as in we are trying to study cell division, or say growth of the cell, or cell differentiation in terms of maturation of the cells, or things like that. How, as we develop with respect to age, we are developing from the zygotic stage to progressing towards the maturity, and then we are going towards adulthood. So across these stages, how our cells are differentiating, how our cells are, that cell behavior, that cell study of the cells is totally different across these stages. So therefore it is very important to understand this differently across different stages of human development. Okay, so therefore, to study and to get a bigger understanding of applications of human genetics, we need to understand these processes, which involves a very detailed and a very minute understanding of cell at the level of our cells, or the molecular level, or even at the biochemical interactions that we generally come across, right? So genetics is all about we know we are directly communicating to the population, or we are talking to the population, but before that we need to have a very baseline understanding about the cells, about the molecules, about the biochemicals, different form of biochemicals. Then therefore all together in a segregated way we can approach towards medical genetics. Okay, if we go by few of the evolutionary phases of how these things evolved, like we have seen Gregor John Mendel's experiments with bees, how he recognized, through his discoveries, we were able to identify the at the level of genetics that we can explore ultimately. Then there were different discoveries simultaneously going on in terms of identification of hemoglobin, or say identification of sickle cell. And over the period of time we're able to identify that what is the genetics behind the developmental challenges, or say the chromosomal abnormalities associated with the development of a child. For instance, we can talk about Down syndrome. So all of these advancements have also led to the growing interest of technical innovations. But before that, again, we need to have these minute details about what is exactly happening at the molecular level. We know Down syndrome is happening because of the extra chromosomes, and therefore these these uh, innovations, or say these discoveries, have happened only when they were aware that at least these are some standard guidelines, and like serving as a baseline knowledge to have help them out with the discoveries, right? In any context, I mean, not just with the Down syndrome, anywhere. So all these advancements and discoveries therefore have a predisposed baseline knowledge about these molecules, about these cells, and about these chromosomes, everything. So therefore it's very much important to understand cytogenetic method in that context. And specifically when we are talking about medical genetics, see, we have talked about the historical background of medical genetics. Evidences state that it evolved in almost, at the end of 19th century. So it started by then, and over the period of time we have progressed with the techniques and technologies. But you can see with the advancements we have reached in 2000, we were able to we had the entire human genome in front of us. So all these discoveries, therefore it's even if we talk about these lifestyle disorders, earlier what used to be the case was we specifically talked about single gene disorders, or the knowledge was strictly limited to one gene, one cause, and one effect sort of pattern. But now we have multifactorial diagnostics, like even for the lifestyles. Even in case of prenatal diagnosis, we have seen how different exposures of or different factors can lead to one specific condition. So these multifactorial conditions, diagnostics of these genetic disorders which are having predisposed multifactorial conditions, is again the outcome of these baseline knowledge. Again, we need to have that cellular biological baseline study about what is the the cellular composition, and at what stage can we study, and what stage can we expect the structural abnormalities generally with respect to any genetic disorder specifically, right? So all of these therefore become important if we talk about cytogenetics. Most of the diseases have also a probable genetic as well as the environmental basis. So the genetic components, which may be the major one, right? But in some cases it is notable that it's only 50% of the cases that it's because of the genetic predisposition. And in most of the cases, even today, we cannot have a clear explanation ki okay, this is the exact cause for any genetic disorder, because most of the evidences are still obscure. Okay, so therefore clear explanations for every single genetic disorder is clearly not possible. As I in my previous sessions also I've talked that there's a documentary on Netflix on diagnosis, I guess that's the title. So that again very much clearly depicts how different different conditions, they do not even have a name to it. Forget about the diagnostics, forget about the symptoms. People normally are sitting today and tomorrow they had some weird symptom which is not really associated with one given condition. Those symptoms are overlapping with multiple conditions, multiple issues which have a genetic predisposition, but they do not exactly delineate what exactly is the condition. So these symptoms are overlapping with different conditions. So therefore we cannot clearly depict one cause, one effect sort of pattern. Still there are multiple causation, multiple factors responsible for one given condition. So and therefore studying more about the cytogenetics or the molecular technology so far, all these studies have altogether opened up ways or modernities towards biology, and therefore the significance that we can lay upon cytogenetics or the molecular technology is very much helpful in medical genetics per se, like even in case of prenatal diagnosis, which help us obtain the genetic information about an embryo or a fetus. Although there are treatments available to those effective those diagnostics during the prenatal phase, but most of the and technical interventions are still not possible to every single person who has been detected with a prenatal sort of screening with diagnosis, right? So these therefore conditions need to be further understood, further researched upon using these cytogenetic evidences. There needs to be more technological advancement about these cytogenetic methods, molecular methods, but so far we have been using the classic methods. There are few innovations now and then, but coming across all these discoveries, all these advancements, we can say that the ultimate need therefore is to understand what are these cytogenetics, what is the applications of be it biochemical or molecular methods, how these studies are being applied in the field of medical genetics. So these methods therefore becomes very much important, and this background was very much important to be given, because cytogenetics, we know that it is something associated with the cell or something with the chromosomes, but why is it important to study in terms of medical genetics? Therefore you need to understand where exactly is the applications of medical genetics, and how and when and where does what form of knowledge is being required, right? So if we are talking about chromosomal abnormalities, definitely it comes with respect to the cytogenetic methods. Here we do not have really the application of molecular methods or say the biochemical methods, because we are specifically delineating the chromosomal patterns, be it numerical abnormalities or structural abnormalities. So the cytogenetic understanding therefore becomes very much important. So we'll be talking about this in detail in this and the subsequent sessions. This session I'll restrict myself to giving you a brief background about what cytogenetics is. In the next session we'll be talking about the techniques of the cytogenetic methods exactly. Like we'll be talking about different binding techniques or fish method. All of these will be coming in the subsequent sessions. So giving you an overview of what cytogenetic is, basically it's understood as a branch of genetics, and in some situations also a branch of biology, or in medical situations, in medical discipline, you say it as a study of human anatomy, right? Cytology, cell biology, right? So therefore it's generally considered as a branch of genetics. Now more or less we see cytogenetics as concerned with a few things, like the cell behavior, or by the role of chromosomes, especially talking during the phase of maturation or during the phase of cell divisions, and thirdly about the chromosomal abnormalities, right? Now when we study these chromosomes and we try to analyze these chromosomes, that science or that study is called karyotyping. We'll be talking in detail. This is called karyotyping, and here what we do is we try to we have set off all the chromosomes within a human cell, and then we try to arrange it in an order, wherein we locate the centromere on one given line, and then we the shorter arm, that is P, is above that line, and the longer arm, that is Q, is below that line, and that is how we arrange these chromosomes in an orderly fashion. And at the end we have two sex chromosomes altogether located at the end of this karyotyping. So these is how these patterns basically helps you understand if there is any abnormality visible, or if there is any numerically if there is any abnormality, or structurally if any chromosome is showing some deviation from the normal visible chromosomes. So therefore again they helps with you with the chromosomal abnormalities. So again there are different techniques, like we use the banding techniques, we have stains to highlight the chromosomes, and then we study respectively different banding techniques, right? So that again is one of the method. Now coming on to a few evolutionary or some historical evidences which speak of cytogenetics as a as a evolving science. It's not wrong, or it could not be incorrect to say that it originated very very long back. It's almost century old discipline, but now even today it has its applications in lot many disciplines of biomedical sciences, or even the medical sciences, even in interdisciplinary sciences we can talk about it. For instance, in Delhi University there is the coursework on cell biology along with the major discipline of anthropology. So it's an optional subject, elective paper for 1 or 2 semesters. So therefore these cytogenetics is also academically being used interdisciplinary for understanding, because biological anthropology also talks about the cellular approaches, and therefore cytogenetics or cell biology becomes very much important to be understood. These techniques therefore we are studying otherwise also, but cell biology as a paper helps you understand it more specifically. You are approaching the cellular understanding, right? Then in 1888, Fleming, he published the first illustration of chromosomes in his publication. Then in 1888 again, Valdier, he was the first person who termed this thing as chromosomes, what it is and what exactly is the chromosome. Then in 1912, Hans von Winnewater, he reported different chromosomal numbers during the process of spermatogonia and oogonia, as in 47 chromosomes were observed by him during spermatogonia and 48 were observed during oogonia. But later on there were different discoveries. They said that this is incorrect, or they reported their observations, and that was correct. But he was the first person who reported altogether. Now in 1956, now when we have identified the chromosomes, we wanted to study the chromosomes and their deviations across different persons, right? So as and when medical records were coming up, we were trying to study how chromosomes are structurally showing deviation or numerically showing the deviations. So there was this discovery of ornithine dye, which was used to stain the chromosome so that the banding could be conducted, and using that banding technique they were able to locate or orderly arrange the chromosomes based on the size or the position of the centromere. Centromere position again varies, as in with respect to age, also with respect to cancerous conditions. We'll be talking about it. So altogether these dye was helpful in the banding technique. Later on they identified that maybe there were some technical issues. Now we had the fluorescent microscope, which could help us identify the brighter, the darker regions of the chromosomes. Therefore another dye which was obtained in 1916 was the quinacrine dye, which again is used to stain the chromosomes, and therefore these microscopic advancements again helped us identify the bright and the dark regions, which are called as the Q bands. Therefore altogether these banding techniques was used. Just to give you the introduction of banding technique, basically they are used so that within one given set of chromosomes we can identify whether there's any structural or any numerical change. Again in the karyotyping also we observe this. We try to observe if there is any structural change in the chromosome, or there is any increase or the reduction of chromosomes in a given set, and altogether we will try to assess whether there's any chromosomal abnormality in the given sample or not. Okay, so altogether there were different banding techniques emerging. With respect to that, there were new dyes that were coming up, because dyes play a very crucial role in staining one particular region of the chromosome and therefore observing it under the microscope. So that was the approach. Then in 1929 to 1938 there was an American cytogeneticist. His name was McClintock. He was the one who has been for a long time conducting the researches on, like back then, he was conducting researches on chromosomal aberrations. So therefore idea of having deviations from the normal set of chromosomes was coming up, and therefore chromosomal abnormalities, chromosomal aberrations, which recently is a trend in genomics, in cytogenetics. Not a trend so to say, but an area of interest of research, a large area of interest for research. Has initiated back in 1929. So he was constantly working based on medical records. He was constantly going across different chromosomal aberrations that were coming up to him, and he was trying to study all of these chromosomal aberrations. And over the period of time there were different geneticists, different different disciplinary approach to understand these chromosomal aberrations, not just limited to the cytogeneticist, but also to different medical as well as interdisciplinary fields. Now, the basic principle for cytogenetics is to examine and analyze all the 46 chromosomes, the set of chromosomes that we have, to obtain them, to observe them if there is any change numerically or structurally, right? I mean, for different chromosomal abnormalities, I have made another video. You can just brush up your knowledge. I'm pretty sure you have seen that video. Different chromosomal abnormalities, structurally, numerically, polyploidy, aneuploidy, many different abnormalities. So all of these have to be observed, they have to be obtained, they have to be understood very critically, and in fact there could be some deviations from the standard abnormalities that we have observed. So that could be one, again depending on whether the condition is severe, whether the condition is profound, right? I mean, the phenotypic condition I'm talking. The genotypic condition definitely have a certain chromosomal predisposition, which so far has been, I mean, as per the documented records, we can check to whether this condition is specifically talking about one genetic condition, or one chromosomal change or numerical change or structural change. So that ways we can relate it. Okay. Now, one technique that has been globally used, or so-called the gold standard technique used across the cytogenetics, even across other disciplines, is the technique of karyotyping. Now, I've already talked about what karyotyping is. It's the arrangement of these chromosomes in an orderly fashion based on their size and the position of the centromere, and the sex chromosomes are located at the end of that karyotyping. Again, we are observing the structural and the numerical changes in this in these patterns. We'll be talking further in detail about karyotyping tomorrow on the next session, but this is just for the introduction. Okay. Now, there are two kinds of cell divisions we are already aware of: meiosis and mitosis. Mitosis basically is a somatic cell division which is responsible for growth, or say tissue differentiation within your body, or proliferation, and many things. Altogether, it's the process that we observe during the mitosis phase. Second is the meiosis, which is specifically responsible for the production of your gametes. So these are two different cell divisions. Now, chromosomes are not visible under, when we study these cell divisions, we study whether there is any procedural change during cell division or at the outcome of the cell division. So in case the chromosomes are increased or decreased in both the male and the female chromosomes, so that varies, and therefore that has an implication over your next generation. So now, when we observe these chromosomes, these are not visible under a light microscope, especially in the non-dividing phase. These are not visible. Non-dividing phase as in the interphase cells, if I'm not wrong. So that is the phase when these are not generally visible. As in when these cells begins to divide, there is a thread-like structure called chromatins, which begins to condense during within your nucleus, and it is visible later when it reaches towards the metaphase stage. Only then we can identify, and then we can do the proper banding and staining of the chromosomes, only when it reaches the metaphase stage. Before that, these chromosomes are not clearly visible, and therefore there's no point banding or karyotyping them. Okay. Now, a part of chromosome, which is generally called the centromere, is the primary constructions which separate the two arms of the chromosomes, which is the upper arm, that is generally the shorter arm, referred by P, small p, and the longer arm being the in the lower half of the centromere, that is the Q. And this P and Q basically is if I'm not wrong, it's a Paris nomenclature used. So that is how we refer or label the upper arm, the shorter arm, and the longer arm. Now, a centromere, as we are talking about this component of the chromosomes, they within themselves they comprise of repetitive DNAs, which is responsible for the separation of chromosomes during the process of cell division, right? Then each chromosomes have the chromatids, identical strands, or the sister chromatids as we refer to in the layman term. This is visible after the S phase of the cell cycle. Okay. Now, next part of the chromosomes is the telomere. Basically, these are the most distal end of the chromosome. They are helpful in two ways. Firstly, they are helpful in the process of chromosomal replication. Secondly, they maintain the structural integrity of the chromosomes, and it sort of seals the end of the chromosome so that no further replication could take place, right? Now, when the cell divides, basically the process during cell division, it's the one arm keeps getting shorter, and telomere therefore keeps getting shorter. And there comes a time when there is no further division possible. It is the time when this telomere is extremely small. It cannot really support further division, and therefore total termination of the cell division takes place. It is generally observed when a person ages, or when he or she is in a severe cancerous stage. So that is the time when further division of that particular cell is not really possible. So these chromosomes parts equally play a very major crucial role during the process of cell division, during the process of maturation in across different phases of human development. I'm speaking. So every different stages have their specific role in development of these chromosomes, representation of these chromosomes, the representation of different parts of chromosomes. Okay, altogether, therefore, these parts needs to be individually studied, individually if I talk from the research point of view, they have to be individually hypothesized with respect to one given condition. So altogether, they are very much important in the cytogenetic method, in the cytogenetic studies. Okay. Now, based on these location of centromeres, we classify these chromosomes into 3 or 4 categories basically. One is the metacentric, wherein centromere is located almost or near to the middle of the chromosomes, okay, near to the midpoint of the chromosomes. Then there is submetacentric, that is somewhere the centromere is located, or it's quite displaced from the midpoint of the chromosome. It's towards one end, or not towards one end, but deviated away from the middle point, not in the midpoint, or not almost near to the midpoint. Okay. Then you have acrocentric, as in your centromere is almost towards one of the extreme ends. Okay. And fourth is telocentric, wherein centromeres are almost at the tip of one end. Okay, so you can have one telomere itself visible instead of having one chromatid. Okay, so these categorization again help you identify what is the structural abnormality, or if there is any deviation from the from these particular visible locations of centromeres, and therefore then you can observe if any genetic condition is associated with those locations or positions of the centromere. So as I was speaking in the previous slide also, all these essential components of the chromosomes have equal contribution in their association with any form of chromosomal abnormality. Researches are therefore conducted in terms of altogether we can see, or with respect to one given component we can highlight that okay, this association with, for instance, centromere, if it's totally deviating from these 4, we can say that okay, this condition is associated with something different from what we see in a conventional sort of locations of centromeres. So therefore these classifications needs to be clearly understood. Now, chromosomal abnormalities, basically these abnormalities are understood as the error in the chromosomes, or the number of chromosomes, also the structure of the chromosomes, which arise during the development of an egg or a sperm cell, right? So both ways, like the composition of that cell or the number of the chromosomes within the cell, all together they are responsible for or leading towards a chromosomal abnormality or chromosomal aberrations or chromosomal disorders. So anyway, there are different terms used for it. Unlike other cells, basically the sperm and the egg cells, they have only 23 unpaired chromosomes. When they unite during the phase of fertilization, they ultimately leads to or results in the formation of a fertilized egg with 46 chromosomes. But if there is any error during the cell division process itself, it results in an embryo with a sort of chromosomal abnormality. Okay, so if we have to speak of any common abnormality, I mean, let's say about Down syndrome, wherein a person is having 3 copies or an extra copy of a chromosome instead of what we have conventionally, the 2 specific copies of that chromosomes. So therefore that deviation from the number of the conventional chromosomes present within one cell is deviated, and therefore that leads to a chromosomal abnormality. Now we have 2 main types of abnormalities. I mean, I've specifically talked about these aberrations in a different video. I'll try to attach the video link in the description, so just go through that video if you have any doubt about what are the 2 different forms of abnormalities. First is the numerical abnormalities, as in when there is incorrect division and they have the egg and the sperm cells, they have an extra or a reduced number of chromosomes within themselves. That is a situation generally what we refer to as numerical abnormalities. Numerical as in number wise there is a deviation. Then there is a structural abnormality when there is the alteration of the structure of the chromosomes, as in within one chromosome, within a pair of the chromosome, one of the chromatid gets deleted or one of the area gets deleted. Then there is a duplication of the region of the chromosome. Then there's translocation, inversion of the chromatids. Then there's a formation of ring or isochromosomes. There are different structural abnormalities so far we have come across, and many new innovations are constantly happening about it, right? So this is about the numerical and the structural chromosomal abnormalities. Please go through the aberrations video if you have any issue with the aberrations. Now question is, how do we detect these chromosomal abnormalities? Slowly we'll be proceeding towards the methods, okay? So chromosomal abnormalities basically are diagnosed at multiple phases, as I already spoken, that across different stages you have different ways to approach abnormalities, you have different observations towards these abnormalities. So therefore during birth or after birth, if we say, we can identify using the prenatal diagnosis methods. There are different prenatal tests we have already talked about, invasive test, non-invasive test, amniocentesis, chronic pulse sampling. Different tests are done during or prenatally before the birth. So different ways we can detect the chromosomal abnormalities at different stages of child development or human development. Now what we do is basically during the sample collection, the cells are collected or cells are obtained, and they are grown within a lab-based setting. From these cells, the chromosomes are obtained. They are observed under the microscope. Using the staining, we do the if we have visible structural change or visible phenotype of that genetic condition, then probably we'll do the staining method. But apart from that, if we want to see whether there is any genetic deviation from the chromosomal deviation from the conventional sort of visible chromosomes, then we'll do the karyotyping. A picture or a orderly arrangement of these chromosomes is done. They are arranged from the largest to the smallest size. Then the karyotype shows the number, the size, the shape of the chromosomes, and therefore they help the experts to identify any form of abnormality. Also, there are other methods like FISH, fluorescent methods, which also helps in identification of the chromosomal abnormalities. Again, it depends whether we are aware whether the person is having the chromosomal condition depending on the phenotypic expression or not, or in case of any late age onset or in case of any prenatal diagnosis, we do not have the phenotypic expression so far, then we do the genetic expression using the karyotyping. The term karyotype basically refers to this display of chromosomes of the cells by lining them up, beginning with the largest chromosomes and with a short arm which is oriented towards the top. Okay, we generally put P above that hypothetical line where we put the centromeres. Okay, in humans we have seen that there are 7 A to G groups of autosomes which are recognized. There are 6 chromosomes that are placed all together at the end of the karyotyping. Here you can see X and Y is almost located at the bottom of the karyotyping. Then there's a diagram of the karyotype based on chromosome measurements in many cells, which is generally referred to as ideogram. So when we take that picture, we upload it. That software is used to click a picture of that karyotype. Manually also, for practical purpose we do it, but generally we use the software, and therefore it's that picture is called as ideogram. Now in this picture, in this diagram, the chromosome number is 13, that is A group, they are metacentric. Chromosome number 45, that is B group, are metacentric. 45 and 612, that is C group, are submetacentric. Okay, that was with respect to this picture. But again, this karyotype basically indicates the it depicts how different size wise these chromosomes are located, how and at the end of these depiction, at the end of this karyotype, we have the 6 chromosomes. And using, when we do it manually, what we do is hypothetical line is drawn, for the practical purpose, a hypothetical line is drawn. Using the centromere, we put it on that hypothetical line we see, which is a small arm we located above the that line, and then the longer arm below that line, and that is how we do the karyotyping. So basically it's again depicting if there's any structural change or not. So for the further depiction of karyotyping, different banding methods, I'll be having a separate session. Okay, so this is about chromosomal abnormalities. Any question, any queries, any feedback is welcome. Thank you.